Okay, so you're probably wondering what I'm doing with all these jars here. And that's a good question. Essentially, we are about to do a liquid culture test to see what types of populations of microbes that we can create. Looking at either grasses, soils, leaves, it is looking into the rhizosphere, which is more of the below ground, phyllosphere, which is actually on the leaf material, and then also essentially based on the sugars or the levels of waters, or if the rice, what we're trying to see is based on the carbon that we are feeding them. If it is a more complex or simpler carbon, we should see different populations, as well as the water level being either a high or a mid. We should start to see more anaerobic or aerobic conditions. So you're going to get some sugar, some rice, and everything in here, cap them up, and then go for about 24 hours. We're adding about a teaspoon of each in the rice and the sugar. Rice. A little bit more on that one. Rice. And then lastly is to be filling them all up with the water to actually kickstart the biological process that's going to be going on inside each of these jars. So it's going to be middle for these three, and then all the way at the last one is going to be almost entirely filled, be creating an anaerobic condition. So we're going to go ahead and fill these up with water. Okay, and there we have it. We have four different various jar samples that we gave different levels of water, different substrates to actually collect hopefully different rhizo or phyllosphere microbes, and as well as a simple versus a complex carbon to feed either a simpler or complex creature. And in about 24 hours, we'll start to be able to look at these under the microscope, seeing what's going to come, and the longer they go, different the population gets.